Okay, I'm gonna try and do something a little different this time. Um, this article is Cell Physiology, The Changing Color of Fat by Brian Owens, publication date 2014, Nature, Nature Outlook. The different functions of white, brown, and beige fat might yield new targets in the fight against obesity and metabolic disease. When you think of fat in the human body, you might picture a homogeneous white substance, much like a block of lard. But researchers are learning that the role of fat in metabolism changes depending on where it is in the body and even on the type of fat cell. Soon, these differences could be harnessed to fight metabolic disorders such as diabetes and obesity. Let's see something. So that picture wasn't what it wasn't defined. I'm searching fat cells ultrastructural. Ultrastructural means electron microscopy, and that does appear to be what that picture was. Well, this is another like pop science article without defining what the picture is, but this is an article. I'm not going to click on the link because it's the source is ResearchGate, which is like where the dumbest of dumb shill their research. Doesn't mean that the research on there is bad, but it's just purely I'm just not going to click on it. But yeah, it looks like that's the ultrastructural. So scanning electron microscopy is what SEM stands for. There's scanning and there's transmission. Scanning creates just really cool images overall. But there's a good one. So let's just look at this. Just want to see this. Don't know if I'm on my VPN, so I might. Looks like I have access to this article, anyways. So this would be an light microscopy. I can tell just by looking at it because I've been doing this stuff for 20 years or so. Not with fat. I'm a molecular neuroscientist. I guess maybe I could have an intro on this video. So this would be the nucleus, and this is the nucleolus, and then are these representative image of perfusion fixation, quality of needle biopsy of diseased human liver. So these are just cells. In a fat cell, you'll see just multiple droplets or like just a huge storage space, pretty much. Yeah, this is either a multinucleated cell. I don't know much about the liver. Again, I'm a neuroscientist. But there's no nucleolus. Interesting. Anyways, what were we going to? Fat accumulation causes necrosis of liver parenchymal cells at the single cell level. Low magnification of jet fixed needle biopsy of steatotic human liver. So this would be like a fat cell. See how it's just that huge droplet. So the cell is like around here. Everything's kind of pushed to the outside, and then the droplet is in there. It's just interesting, but we were going to this one. Let's see what C is. Inflammation at the single cell level, transmission, electron microscopy picture, so that's an A. And then this one is scanning electron microscopy. Both ultrastructural imaging is usually what I search. If you just search or scanning electron microscopy or ultrastructural SEM, space SEM, you'll see just cool pictures of a lot of different stuff. It's used in engineering to see like where stress is occurring. It's a pain in the ass to do. I've never worked with it, but where I did my degree was in Chicago, and the L, which is the train there, went right over was the red line, went right over where we were. And I know some other grad students who worked with electron microscopy. They put it in the basement. They tried everything to stabilize it, but it was just a huge pain because it constantly had to be calibrated. Isn't the word? It constantly had to be calibrated, but all microscopes you have to calibrate when you're doing, scientific things. when you're doing science. Well, with these, I guess if you're just taking pictures, it's a different step of calibration. I did a lot of measuring under light microscopy, so definitely calibration was like a daily thing. But it would just adjustments would have to be made. The tech would have to come out a lot to mess with it, and yeah, I don't envy people who do this. Okay, C, scanning electron microscopy, picture of an inflammatory fat follicle showing the uptake or release of filaments supposedly being Mallory substance. We would have to read this article to know what Mallory substance is. Anyways, I just wanted to see what the point was. I just wanted to see what this image is. So this is likely scanning electron microscopy. It's probably, it's definitely falsely colorized because every time you have images, they're in black and white, right? Unless you, I mean, you can immuno. How do I get back to mine? Let me do scanning. I don't know if you probably want to do it for scanning electron microscopy. Let me do ultra structural immuno labeling. You don't get these vibrant colors with it though, so this is all done after the fact. Well, these are actually words, so you don't immunolabel and find words on tissue. I was using a laser dissection tool, and I wrote my name one very late night in a slab of brain that I didn't need it anymore. It wasn't a human tissue. I feel like it's disrespectful if it's human tissue. I mean, it's disrespectful anyways, I guess. It's fun to play with the tools, but you have to think about like a life was given for that research, or I try and think about that. So you can immunolabel things, but the coloring, anytime you see coloring like this is done after the fact. And this is also, I would hazard, just definitely a modified image. They still should have put some details under here, but they probably didn't know what they were looking at because it's just Getty images. They probably just searched like brown fat cells or fat cells. Okay, back to the article. The predominant form of fat in mammals, including in humans, is known as white fat. White fat cells are found throughout the body, primarily under the skin, as well as in larger deposits of the abdomen. They are highly specialized for energy storage, hoarding calories in the form of lipid droplets. And again, that's what we saw with the microscope fat cell. So here I missed the old Google Images thing, but here this is like the cell. This is just a gigantic fat droplet within the cell. This is labeled. So yeah, the cytoplasm, everything is pushed to the outside. Mitochondria, nucleus even, you see the nucleus is here, and then that ring around the cell, and just a gigantic globule. In fact, I have something I was working on. That's a nucleus, doing this heat map. So this is a motor neuron, and you can see the nucleus, sorry, the nucleolus is here, and then you have the cell, versus the fat cell, where everything is just pushed to the outside. Neuron is a very specific cell type, so maybe not a great example, but that's just something I was working on for a study. Okay, back to the article. The predominant, isn't this better though than just looking at a glass of liquid? Hopefully the microphone's working. My speaker isn't working, and I have no clue if this microphone is working actually, because I have no way to listen to what it's recording. It should be, oh well. I guess I should check that now. Hang on. No. Never mind. I did want to comment that, so my degree is in molecular neuroscience, although I often joke that the degree itself just says Doctor of Philosophy, and it could be in anything. But I did my degree at a medical school, and we had to take these physiology classes that I wasn't particularly interested in and never would have learned stuff otherwise. Learned stuff about, other organs. And in terms of fat, pretty much brown fat you just learn about with infants. So infants are born with a large amount of brown fat, and that insulates them. Um, and I don't think I really knew brown fat was a huge component of adult human. I mean, it's not a huge component, but was a physiological component of adult humans until recently. I came across a video where it was talking about a research study. What was it? Oh, someone was saying to sleep sleep in cold temperatures, like 67 degrees Fahrenheit or something like that, and you'll burn fat. 67 degrees Fahrenheit to Celsius. 67 degrees Fahrenheit or 19.4, 19.5 degrees Celsius, and you'll burn fat. And then it was actually a study talking about brown fat, and it was just kind of a stupid thing because anyone sleeping at 67 degrees or is going to use blankets was my first thought. But yeah, and the idea of burning fat while sleeping, like the whole study was looking at brown fat, which is not what you do cardio to burn. I don't think. Let's keep reading the article. Let's let someone who knows about this talk. Back to the article. The white cells are complemented by brown fat cells, which specialize in converting chemical energy from glucose and lipids into heat, generally when an animal is exposed to the cold. This burning of carbohydrates and fat also helps counteract obesity and metabolic disorders. But, says Patrick Seal, a developmental biologist of the University of Pennsylvania in Philadelphia. So this isn't a regular scientific article. Nature Outlook. Kind of reads like a pop press article. It's probably a step above that. But usually you don't have interview quotes. Not in stuff I read. I guess maybe this is what the Nature Outlook journal is. Nature has a bunch of smaller journals, like Nature, just Nature is like a huge journal that everyone wants to publish in because the impact factor is high. I don't prize the articles from there highly for various reasons, but they have a bunch of small other journals, not necessarily smaller. So Outlook looks like this may be not editorials exactly, but how would I even say that? This is gorgeous. It's not a review, not an. I guess editorial would be the closest thing, because you're gonna get an overview of a topic, but you're also gonna get the coloring of the author, which with a straight review, you get a little bit of that, but ideally not a whole lot. But you can see here, even just with these what's it called? Not figures, but not thumbnails. I can't think. It's morning. You can see that these are geared towards clicking towards the common person, like, oh, what are these faces, or oh, why is this scary face here, and scan research, oh, I want to click on it, which, like, for me, I mean, this might be interesting if I was doing dermatology, but like this other stuff, like if I'm looking for something scientific, these mean jack shit to me. But I can see a pop person, or I mean a common person, just being interested in clicking. So that's, now we know what Nature Outlook is, and that's what this article is from. Doesn't mean it's not real, it just means it's not it, I don't want to say not scientific. It's not a scientific article. So scientific article would be original research, review, etc. This is something other. Anyway, it doesn't mean that the information isn't good, is what I was trying to say. Back to the article. But, says Patrick Seal, a developmental biologist at the University of Pennsylvania in Philadelphia, that does not mean the two types of fat are heroes or villains. Often one portrays white adipocytes as the bad guy and brown adipocytes as the good fat cell. He says that's not really the case. White fat cells serve as an important role in protecting against metabolic disease, but the key is to keep those cells healthy. So I'm scoffing because anthropomorphizing molecular biology or biochemistry, physiology, anthropomorphizing anything that isn't human, that isn't anthro, is asinine, might be too strong, but it's not, it's just stupid. it suggests to me that you don't fully understand what you're working on, but that you're used to talking to lay audiences and getting positive feedback from talking to lay audiences, which then it makes you dumb down stuff more, and in the process, you get dumbed down. Like, you talk to a kindergarten teacher who talks to kids all the time, doesn't mean they aren't smart, but they're used to talking to a certain level of child, and that comes through in their speech with adults. I feel like scientists are the same way. When you anthropomorphize stuff and like, hey, this is groundbreaking, but white adipocytes are not the bad guy. Brown adipocytes are not necessarily the good guy. They both do good and they both do bad. Like, no, it's just fucking shit. Like, for science. But speaking to a lay audience, I could see applause occurring at that. Anyways, that's why I was scoffing reading that. Back to the article. Healthy white fat protects the body by providing a quote unquote safe home for lipids, which can be toxic to other tissues such as muscle or the liver. So these fat cells hold on to lipids until the energy they are restoring is needed when they release them into the blood. Again, scientific article. I mean, you see it, but starting a sentence with so is just an interesting choice. So when they talk about lipids being toxic, I think they're just reactive, meaning stuff binds them, changes their structure, and they in turn stuff, highly scientific word I know, and they in turn act on other components. I believe that's how lipids are. Instead of toxic, I would put the word reactive. Back to the article, when functioning appropriately, white fat cells will limit metabolic disease. Where white fat cells are located also matters. Subcutaneous white fat scattered throughout the body in depots under the skin is not associated with metabolic disorders and may actually be preventative. It is only the abdominal white fat that, when it gets out of control, can cause health problems. And at Seal, people with too few fat cells, such as those with a condition called lipodystrophy, have many of the same sorts of problems as people with too much fat. When they are functioning appropriately, white fat cells will limit metabolic disease, he says. They are actually a protective cell type. And again, that's by sequestering the lipids. I mean, lipids are everywhere. Like, the cell membrane is lipids. So just saying sequestering lipids is ridiculous, because everything in the cell, going back to, like, this is lipids. This is a double bilayer around the nucleus. Everything is lipids. Not everything. Everything is protein. But lipids are everywhere. So this is talking about, would it be triglycerides? I think so. A specific form of lipid. Problems arise when white fat cells store too much lipid. They begin expanding and proliferating rapidly in a process that resembles the growth of a solid tumor, says Philip Scherer, a cell biologist at the University of Texas Southwestern in Dallas. The blood supply cannot keep up with this expansion, and the cells begin to suffer from lack of oxygen. This hypoxia tracks the protein HIF-1 alpha, which I believed it is hypoxia-induced factor 1 alpha, which in fat cells stimulates the extracellular matrix surrounding the cells, leading to fibrosis. The huge oxygen-starved fat cells do not have enough room to expand and get squeezed to death, releasing their lipid cargo. So first thing to note is this is likely talking about an experiment in cell culture. And here it says, resembles growth of a solid tumor. One of the ways to limit cancer growth is to stop vasoneogenesis, the growth of new blood tissue. So the blood supply cannot keep up with this expansion. I'm not sure that happens with solid tumors, outside of my wheelhouse, I guess, but also detection of tumors, neoplasms, is often through the vascularization, because you have, along with the cell growth, or cell reproduction, you have increased blood supply in these smaller vessels. Cancer, is it called neovascularization? Is that the word? You just have blood cells that go throughout tumor, inclusive invasive, can't read that. That's good. Wait. Neovasogenesis? That that's not the word. But anyways, the way I know it's in cell culture, the blood supply cannot keep up with this expansion, and the cells begin to suffer from lack of oxygen. All of that stuff is stuff you would know from cell culture. I mean, it could be a biopsy, but this really sounds like cell culture work, which doesn't mean it's not real, but it does mean that the physiological standard has not been set. This hypoxia attracts the protein HIF1 alpha, which again, that's something you learn through cell culture, or you verify through cell culture. They could have found it physiological from biopsies and then verified it in cell culture, which in fat tissue stimulates the extracellular matrix surrounding the cells, leading to fibrosis. So yeah, all of that sounds like cell culture work. Let me double check. These open. HIF1 alpha. See if that'll go. So this is an symbol I use for primary gene resource because they update every 6 months or so. And then I have Unipro right here for proteins, but hypoxia-inducible factor 1 subunit alpha, as I suspected, unless I said something otherwise, in which case it's not as I suspected. And in this page, you get the gene name. I guess if you're looking for a certain gene name and you're a layperson, you would want to go to Hugo gene nomenclature. Ugh. And this is, so on this page where you have source HG and C, and then the symbol, that means that this is the source. So HIF. I don't know why I typed alpha. I should have known it, but and then you get gene card, just like a card, you know, like you picture. When I was doing wet lab work, I actually had a box, recipe box. I would use stuff used in cooking because then I could protect it from fluids. So I actually had recipe cards where I would write down protocols, and then they would be sleeved in like this plastic sheathing. And then for book covers, I would use cookbook recipe covers. They're like plexiglass covers. But so this would be like a recipe card to me. But anyway, a gene card is what it's referred to. And then you see the ensemble like here, ensemble. So you can get the standard name as of whatever date you're searching or whatever date the database has been updated, which Hugo does not have with ensemble. It's always right here. You can see past archive sites. So this one was recently updated. And in Unipro, if we can get this to come up. So there are the Unipro links here. Hi Tex. I'm just going to search HIF1 alpha. You can search the ensemble ID, the gene ID, or the gene name, or you can use those links. So several things come up. Limit to human here on the left tab. And based on, so these are different. We want the one, like this one is probably, and I think this stands for verified. But when you open this up, you'll get the function of the protein. So now we're talking proteins, not genes. Sometimes the same name, oftentimes not. And so you'll get a general function with PubMed reference for where that information comes from. And the Unipro, Swiss Prot, they have these categories. So I download all information, and then I have a pipeline to run it through in Python. You can hire me to do stuff like this. I'm very poor. Please hire me. But their function in the output, it's function CC, and they have subcellular location CC, which is what I'm doing with this. I'm making a heat map for the Parkinson's disease study of expression of what expression of something in a certain cell location does. But this, you can read this function. This is very well curated. That was my point, what I was getting to. It's above and beyond gene ontology, which most people use gene ontology. Gene ontology is useful, but their summary I'm writing several pipelines to extract information from the summary because it's just above and beyond what you can get from gene ontology. You get additional information in determining pathways. It's extremely useful. Same with the subcellular location. But anyways, to look at this page, you would search something, you would find what you're looking for, and then you can read up on it. On the left hand side, you have names, so you have past names that it's been known by. Sometimes when you publish on a certain protein, you'll publish with a name that you come up with, and then later it's determined that that's the same gene as this HIF1 alpha. Or there are several names, and then one becomes accepted, and that's often why you have different different names, alternate names here. But sometimes, oh, not archaic. Yeah, I think I do use the word archaic, or just older names, past names. So gene name synonyms. And part of what I do is I go through studies. I get work, or I get uh, what do I do? Um, I get vast amounts of data from like next generation sequencing studies, and I standardize the names throughout. I use ensemble a lot throughout different ensemble IDs. In some ensemble, I don't know how people say that name. And through different protein names, and then I put all of the information in one database. That way, it studies aren't just one and done. That way, you can compare across studies despite them having different names. So in disease research, it's very important because, like, say someone publishes MOP1 a lot in, let's say, 2005 to 2010, and then that's like a hot gene for a certain disease during that time, and then later this hypoxia-inducible factor 1 alpha is a hot gene, and anyone who studies this gene may know that MOP1 is the same thing, but often the link isn't made. So then you have a whole new body of research on HIF1 alpha, but like what happened to the research with MOP1? And it's very important to do this. The main reason I do it is we tend to have genes of focus, which is like looking for your keys in the spotlight or in the lamplight. You does everyone know that story? So it's a, what's it called? Not anecdote. I don't know, guys. Welcome to my content. So the idea is like a guy leaves a bar, he loses his keys, and then he's looking for them in the snow under a spotlight. Another guy comes, or under a lamplight. Another guy comes up. He's like, "Hey, what are you doing?" The guy looking for his keys is like, "I'm looking for my keys. I dropped them." The second guy is like, "Oh, I'll help you look for them. Where did you drop them here?" And the guy's like, "No, I dropped them over there, but the lamplight is here." Lamp light analogy. And this often happens with genes in disease research. People keep looking at the same genes. Yeah, so it would be like you're looking at the lamplight when really you lost your keys over here. Yeah, I lost them. Lamppost. Yeah, I lost them over there, but the light's better here. It's a perfect analogy for what happens in disease research, and um, having different names for genes doesn't help, especially when they aren't the genes of focus. They aren't the genes in the lamplight in the past or present. So, anyways, looking at this page, subcellular location, very useful. And this is like, I think it's hand curated. I think they have a pipeline they run, machine learning pipeline, and then it might be hand verified. But this is very useful information above and beyond. So there's three main classes of gene ontology. There's biological processes, molecular function, and cellular component. Cellular component would be like the subcellular location, but so here, biological process, cellular component, molecular function. Cellular component would be like where in the cell stuff is located. But with UniPro, this is more useful than that. So, people don't know that, which is good because, I mean, again, I'm poor and need business. disease and variants, so you can see just various publication factors on either SNPs or verified mutations with with these disease indices. So these are going to be protein locations actually. So it would have to be probably a non-synonymous change, or actually it can also be a post-trans or a modification to that amino acid, or post-translational modification, I would hazard. Yeah, natural variant would be something like SNP here, DB SNP. Okay, and then variants, which you probably won't get into as a layperson. A lot of links to other sites, and here we see that HGNC again. That's the Hugo gene nomenclature ID. Post-translational modification processing. So you can go through all of this, but the main things you would look for is probably the name, and the function, which right at the top. Function including the gene ontology. So if you aren't a scientist and you make YouTube videos, that can make your videos more accurate. Back to the article. As the cells start to die, macrophages swarm to the fat depots to try and clean up the mess by carrying away the lipid droplets. The problem is that the macrophages cannot clear up the lipids fast enough, and so they begin to spill over into other tissues such as the liver and pancreas. And remember, we had said in this article, this is me talking, not the article, that lipids are highly reactive, or what did they say, toxic? Let's just use the word reactive, it's better. So when this you can imagine, if we go back to our fat pictures, these giant cells, when the cell starts to die, one of the first things that happens is the membrane breaks, and then the lipid droplet is also enclosed by a membrane. So once those membranes break down, all of this exuding into fat cell dying macrophage, all of that going into the extracellular space, macrophages cleaning it up. You can see how stuff gets away, so to speak. So this is profiling induces metabolic. This might be a dying cell with, I don't know if they labeled resident macrophages. Let's look at this picture. Just the picture, please, no words. Live image of AT explants of HFD-fed green mice. I would have to read the article to know what those stand for. Unless sometimes it's at the end of the caption or legend. Degradation of adipocyte remnants in AT explants occur in 3 distinct ways: efferocytosis, fragmentation, or CLS formation. Okay, this is a whole other area. And chi-fed or high-fat diet-fed animal. So this might be interesting to read. Adipocyte death triggers a pro-inflammatory response and induces metabolic activation of resident macrophages. Not. Never mind. Back to the article. Those tissues form lipid droplets as well, but they aren't quite as good at it as fat tissues to share. This inappropriate accumulation of lipids in fat depots and in tissues that are not equipped to handle them results in continued low-grade inflammation and ultimately in metabolic disease, insulin resistance, and type 2 diabetes. Until a few years ago, it was generally thought that after infancy, humans had only white fat, in contrast to hibernating animals and small rodents in which brown fat is retained. So this is until a few years ago. So maybe in grad school, 2014. Yeah, so that probably was the thinking at the time, was that only infants had brown fat. Which makes me feel less remiss about not knowing that adult humans have brown fat cells. Where are we? Until a few years ago, it was generally thought that after infancy, humans had only white fat, in contrast to hibernating animals and small rodents in which brown fat is retained. In babies, it was considered some sort of strange vestige, a little like an appendix. Not sure that's true. This is Jan Nedergaard, a physiologist at Stockholm University. so it did not attract much attention outside a small circle. I mean, if this person says it's true, it is, because this isn't my area of study. But I mean, babies are effing helpless. They can't do anything. What's it called when those animals can stand right away? Babies aren't that. So I don't know if I would consider it a vestige. Like, it's not something left over that you could do away with and survival would be the same. Babies still need that layer of brown fat. That all changed in 2007, says Niedergaard, when he and his colleagues studied human adults and found brown fat deposits around the neck and collarbone, similar to where brown fat is found in mice. That changed the whole interest in brown fat. He says it became a tissue that could be of relevance to controlling body weight and other things. Not just brown and white. But brown fat is not the end of the story. Scientists had long known that when mice were exposed to cold, brown fat-like cells would start to crop up in their white fat and begin producing heat by burning lipids, glucose, and any other energy-storing molecules available to them. This is not the case of white cells suddenly transforming themselves into brown cells, says Bruce Spiegelman, a cell biologist at Harvard University in Cambridge, Massachusetts. Rather, the white fat depots are seeded with immature cells, which begin to change when they are exposed to cold. But these cells are also not exactly brown fat either. Although they seem to have similar functions and express many of the same genes as brown fat, they are sufficiently distinct to be considered a new type of fat cell, says Spiegelman. Sticking with the color theme, he calls them beige fat cells. And it is not just mice that have this third type of fat. In 2012, Spiegelman showed that most of brown fat in humans is actually beige. Okay, that makes sense. It makes sense because brown fat would be a specific type of cell that would be. The point of it is like a long-term game plan. Whereas these beige cells, it sounds like they're induced rather quickly and used rather quickly. So a higher turnover and metabolism. Because it's saying when mice were exposed to cold, brown fat-like cells would crop up. Yeah, so these cells would then differentiate when exposed to cold versus brown fat, which are, again, like a long-term game plan. Like a hibernating animal would probably have brown fat year-round, deplete some of the reserves when hibernating, and then start immediately after hibernation, start rebuilding those reserves. So that's a long-term plan, right? Back to the article, there's some disagreement, however, about the relative importance to metabolism of the two types of lipid-burning cell. Although Niedergaard agrees that humans have more beige than brown fat, he thinks that, taken all together, beige cells are less able to burn lipids and produce heat than brown cells. The beige fat cells clearly have a lower capacity. All in all, he says, we think the main burning is in classical brown fat, which, again, with what I was saying, like a long-term plan, short-term plan, it's really like these, what he's calling beige fat cells, would be an immediate process to try and keep the organism alive. This is my postulating. Why is this cut off? Well, we'll never know what the top says. Oh, something bad. That could be a very bad word. Not very bad. So this figure says cells overloaded with lipids rapidly outgrow oxygen supply, leading to cell death and inflammation. So this goes back to the guy equating it to tumors. I don't think tumors work like that, but I could be wrong. He did say solid tumors, so maybe solid tumors are different. And also, the friends I have who study tumors are looking at biomarkers and ways to stop the tumors, so that's early in the process, and maybe later in the process it's different, like you don't have proper vascularization, but I don't doubt that this occurs in adipocytes or fat tissue. I just doubt the similarity to tumors. But it's not my area. So adipocyte would be one of these little balls, normal fat tissue, and this is all just this like extracellular matrix. Yeah, I'm gonna call it that. So that's what these lines are. And then hypertrophy is where the cells are getting larger. And I did another article that looked at hypertrophy versus neogenesis, or new fat cells, increased number of fat cells rather. And then obese, and you can see, yeah, this is a pretty good illustration. So it just gets shitty. Like, all this extra stuff comes in, and that's where your inflammation occurs from these cells. And also, once just stuff gets sticky, which it is in a very real electrochemical sense, sticky, you have inflammatory cells and inflammation occurs. It's a mess, basically. So obese, low-grade inflammation, insulin resistance, and ultimately metabolic disease. And these are macrophages, lymphocytes, neutrophils, and other immune cells. And then those are, I guess, the base cells. Oh, reset. Let's see what the top is. Nope, never mind. Disregard. Okay, it does matter. Niedergard is also convinced that brown and beige fat have different roles in the body, and so it is doubtful that beige fat is of any particular interest for treating human disease. But Spiegelman believes that a study he published earlier this year should put those doubts to rest. Spiegelman's team selectively disabled the beige fat in mice by targeting the protein PRD-M16, which is found only in these cells, leaving the white and brown fat intact. So I'm going to pause here and just, because I'm trying to show something, because there's so much misinformation on reaction channels, and this is how you can maybe minimize that if you have the time to do the work. But we're going to look this up. So we want human, we can limit to human over here, but right away we see what we are searching, although this says M16. Histone, lysine, N-methyltransferase, PRD-M16. So if you don't know what that is, which you probably don't, if you're a layman, you would go to the function. And here it binds DNA and functions as a transcriptional regulator, so that's going to affect what proteins are being made. And, several steps removed, but for a lay person. Displays histone methyltransferase activity and monomethylates lysine 9 of histone 3. This is my current study is on a lot of this, but so you could just skip that line. Likely to be primary. Okay, yeah. Functions in the differentiation of brown adipose tissue, which is specialized in dissipating chemical energy in the form of heat in response to cold or excess feeding, while white adipose tissue is specialized in the storage of excess energy and the control of systemic metabolism. And then bisimilarity means that it's been proven for another gene or protein. And bisimilarity, it's usually pretty accurate. Together with CEB, PB, regulates the differentiation of myoblastic precursors into adipose cells, myoblastic precursors. So I may have said stem cells earlier, not quite category. Anyways, that's how you would look up that. Now you know what the name is and what it does. And then I'm curious where the PRD comes in. PR domain, zinc finger containing protein. Transcription factor, MEO. That's how we got the name. Domain means a section of the protein, so that is conserved across different proteins. And then zinc finger, the quick, what you're taught, the quick references, it means it binds DNA. Okay, here. The animals ended up with severe metabolic dysfunction, obesity, insulin resistance, and damaging fat accumulation in the liver known as hepatic steatosis. And this is when disabled by targeting. So it doesn't say what targeting means, but I'm guessing it was either a knockdown or a knockout or inhibition. You don't have to knock out. The loss of beige fat destroys the protective abilities of subcutaneous white fat. Nobody can look at the data and say the animals aren't screwed up when they lose their beige fat. That's genuine scientist talk. I had my main mentor through grad school, and afterwards, I would take him data. I would be like, this is what's happening in this disease these days. So basically what you're saying is these guys are effed, but he would say the full word. And these guys are still effed, but a little less effed. I'm like, yeah, it's pretty accurate. So the animals aren't screwed up when they lose their beige fat. I think nobody can look at the data and say the animals aren't screwed up when they lose their beige fat. Says Spiegelman. I think we have to consider the discussion closed in terms of whether beige fat is important in normal physiology. Okay, well, it's your study, so you, you can close the discussion. And then this figure shades of a cell, three distinct types of fat. Cell have been identified today. Are there more to follow? Question mark. Okay. Brown fat. Shut the fuck up. Are there more to follow? Brown. I was watching us. Nah, never mind. Brown fat cell converts chemical energy to heat to protect against cold weather. Beige fat cell, immature cell, and white fat tissue matures to burn fat. And I guess they could modify this for heat or for as a mechanism of thermal regulation, right? Wasn't that what it was saying? Or maybe just because it could have other physiological relevance that I don't know. Maybe probably the person who made this article also didn't read the research or made this figure. I mean, anyways, white fat cell, most common fat cell used to store fat and found beneath the skin and abdomen. In mitochondria, small lipid droplets, nucleus. Yeah, so here you see this just massive lipid droplet in white fat cells. And here, where the lipids are being used rather than just long-term storage, you're going to have smaller lipid droplets, which makes some sense. Because you gotta, break that open or dip into it to get some lipids out. It's going to be easier to manage and less likely to go haywire if you have the smaller droplets. Fueling interest. Whether the target is brown or beige, fat tissue is attracting attention as a possible way to fight obesity and metabolic disease. Shocker. Researchers are hoping to stimulate the growth of more of the healthful fat cells. If you have enough of them and they're efficiently activated, they can alter the energy balance of the body. Seal says. So the reason why I started doing these reading science articles is I watch while I'm doing my work, playing in the background a lot. I have fat acceptance reaction channels, and that like don't tell Anna O'Brien that stimulating the growth of more helpful fat cells is the way to go. Guys, what you have to understand is to be healthy, okay, first of all, my body is changing. Second of all, to be healthy, she doesn't say first and second, but okay, my body is changing. And while I'm feeling great, ha 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 ha, fake laugh, part of what my body is changing. Oh, wait, I got something here. I'm onto something. My body is changing. I'm lifting weights and I'm getting stronger than ever. And part of that is me putting on tons of muscle and losing all my fat cells. But what current research is now showing is that I have to put on more fat cells in order to feed the muscle. The massive muscle that I'm putting on. Anyways, if you don't know what I'm talking about, it's part of Gorl World, GORL. And if you do know what I'm talking about, insert laughing emojis. It'll make me feel good about myself. Back to the article. The most natural way to do that is through cold exposure. If you said you wanted to build your beige fat, I'd say go lightly dressed in the Canadian winter for a week or two, and you'll have plenty of beige and brown fat, says Spiegelman. He's joking. Half joking. Don't do that, guys. However, I like the Spiegelman guy. I think I would get along with him. However, although some research has suggested that turning down the thermostat might be a good way to fight obesity, any effect will last only as long as the drop in temperature, and exposure to the cold is not really a practical therapy. It would be better, says Seal, if researchers could find a way to stimulate the biochemical pathway that activates the cold response. And that goes back to what I posited, that the beige cells are like a higher turnover, a quick response to keep the organism alive, rather than a long-term process like brown fat in hibernating animals. Back to the article. One receptor involved in this pathway is known as the beta-3 adrenergic receptor. In mice, administering a compound that activates it, an agonist, prevents obesity and lowers the incidence of diabetes. Unfortunately, Cecile, no equivalent agonist has been found in humans, and the search has largely been abandoned by pharmaceutical companies. So I know, I think clenbuterol is a beta agonist. Don't take clenbuterol. But that's used in prep by bodybuilders, to cut fat. Don't mess with your beta adrenergic. Any beta 1, beta 2, beta 3. Beta 1 I think is cardiac specifically. Don't mess with that, because you may be burning fat, but you're fucking up a whole lot of other stuff. Don't, unless a doctor tells you to, don't get into that. Don't do clenbuterol. There are other ways to stimulate the activity of brown and beige fat, however. Well, actually, and going back to this is an important point. So, in research, often we find something that works great. So, originally, I started with multiple sclerosis research, which is, some people think it's a humoral autoimmune disease, humoral meaning antibody based, but it's not. I think antibodies appear later because the system is already so screwed up. But it's primarily an inflammation, autoimmune disease, so T1 cells, and and similar pro-inflammatory condition. And when you can cut off that pro-inflammatory response, you're messing with a whole lot of other stuff. And this happens a lot in disease research. You can find a good answer for something, but it screws with so much else. Especially, I'm a neuroscientist, so working with the brain, it's pretty important you don't generate new neurons. For the most part, the neurons we care about. You don't generate new ones. There is some neurogenesis, but for the most part, the neurons you have are the neurons you have. And if you screw up a system, you can make things worse. So the idea is to make small changes and then let the body, regulate itself and get back on track itself, which is easier said than done. Back to the article. There are other ways to stimulate the activity of brown and beige fat. However, physical activity seems to do it, says Spiegelman. Irisin, a hormone produced by skeletal muscle in response to exercise. Irisin? Can it anything in biology you can say any number of ways. I try and do a standard, so one, a vowel followed by one letter should be a long vowel. So irisin, irisin, unless it's on the end, then it's a short vowel. Irisin, I'm just gonna say it like that though. A hormone produced by skeletal muscle in response to exercise can induce the formation of more beige fat. The hormone FGF21, which is fibroblast growth factor 21, is another promising candidate. Obese humans treated with synthetic version of FGF21 showed improvements in their cholesterol counts and body weight. It is only a matter of time before some of these things become targets for chemicals that can activate the pathways, says Seal. In the meantime, there is still much to be learned about the characteristics of the different types of fat cells themselves, including the question of whether there are any additional categories waiting to be discovered. One possibility, Cecile, is that white subcutaneous and white abdominal fat, which have very different effects on metabolism, might actually be distinct. If you take the cells out and isolate them, they have different gene expression profiles, and they really just look like different cell types, he says. Fat cells might behave very differently and respond differently to different diets and disease states. Spiegelman agrees. He likens the present understanding of fat cells to changing concepts of white blood cells. It becomes B cells and T cells and monocytes and macrophages become dendritic cells, and T cells become regulatory T cells and killer T cells. He says it's entirely possible that within the category of white, we have 2 or 3 different categories, and within the category beige, maybe we'll have 2 or 3 different categories. Yep, checks out. References. So if you want to read this article again, Cell Physiology, the changing color of fat. Also, because I'm doing this, because it just gets irritating seeing a lot of misunderstanding of scientific information in articles. Let me just say, when you cite an article, you don't have to give us the issue in journal, the what's it called, the issue number and the I don't know, whatever it's called, guys. Give the article name, give the journal, and give the year, and then the author. First and last author are generally, for the most part, most important, but in terms of looking it up, the last name of the first author should be enough. When I keep notes for myself, I do the first 2 authors, and then the year, and sometimes the title. So when I keep, let me see if I have something open. I do not. Never mind. Oh, I do have